Great, thank you, Lauren. So I think we've had a number of wonderful talks today, many of which have referred to, you'll hear a little bit later about the clinical trials. So I'll try to, sort of like George did this morning, bring everyone up to speed about the language of clinical trials, what is a clinical trial, why do we do them, how do we measure success in trials. And I thought I would start with a slide that I think is really telling of the way this field is headed. So, grab this. So along this, the horizontal axis here is years. And what I'd like to show here is in yellow is since 1995, the number of publications in PubMed that have gone up exponentially, and the number of clinical trials that are registered on clinicaltrials.gov that have also gone up exponentially in that time. And I think that for researchers, it's incredibly exciting. For patients, I tell patients, you know, I hope in your lifetime you will see benefits of this research that's being done. And I think it's a nice way to set the stage for talking about trials. So I'm going to have four main sections of this. We'll try to go through it quickly, but the basics of trials, we'll talk about what we've learned from recent trials, what trials are on the horizon, and how can you learn about trials in your neck of the woods. So the clinicaltrials.gov is a federally funded website that actually currently, going back to the government shutdown, has a big banner across it right now saying the data may not be up to date given the federal shutdown. But regardless, you can still search on their website for national clinical trials. Their definition of a clinical trial are research studies that explore whether a medical strategy, a treatment, or a device is safe and effective in humans. So really the safety and efficacy are the two main things that are studied in clinical trials. So the types of clinical trials, so this is a perfect segue. Eric just gave us a really wonderful story about preclinical studies. So preclinical studies are those that are done prior to getting to humans, so works in mouse tumors. So what's the next step? So a phase 1 clinical trial tests the safety. We certainly are hoping for efficacy, and there's great scientific rationale usually by that stage. It's often any type of tumor is eligible, usually small studies, 15 to 25 patients, defines the side effects and often the optimal dose for that agent. Phase 2 trials are a little bit bigger. They start to look at efficacy, usually in a specific tumor type. And these may be available nationally. They may just be available at a single institution. Phase 3 trials really start to delve more into efficacy. They're often randomized, meaning one treatment arm compared to another. Often, and if they are randomized, patients don't have a choice in terms of which arm they go on. It's computer generated. Also usually limited to a specific tumor type, and sometimes placebo controlled. I'll comment on that. A placebo is only used if doing nothing is considered a standard of care in that disease. So as Emily Bergsland was talking about earlier, in the pazopanib trial for advanced carcinoid, it is compared to placebo. That's because we don't currently, beyond octreotide, don't have an approved agent in that disease. But the design of that study then allows crossover. So for patients who were on the placebo, they can eventually get access to the drug. And these big phase 3 trials are really essential to assess survival differences. One other comment, I know a question that has come up is the difference between prospective and retrospective studies. So all of these studies are prospective, meaning we have thought about the question, we enroll patients, and we study them moving forward. Retrospective studies are ones that we do with big databases or big data sets where we look back and say, of these 200 patients, what can we learn about them? And those are still important, but for clinical trials, really to get accurate information, we need to study them prospectively. So in terms of the design and interpretation of clinical trials, we've talked a little bit about eligibility. So Dr. Eric Mitra spoke about the PRRT trial and how complicated the eligibility is. And so this refers to going through really a detailed checklist of dotting I's and crossing off T's of figuring out, we want to make sure all the patients are the same, essentially, on a clinical trial. So which neuroendocrine tumors? Should they be well or poorly differentiated? The site of origin matters. Are they growing or not? Have patients had prior treatment? So all of these things go into consideration for eligibility, and every clinical trial is a little bit different. So how do we measure success of clinical trials? There are actually a number of different success measures. We call these endpoints in clinical trials. For neuroendocrine tumor trials, we actually had a meeting that I think George referred to at some point back in 2009 called the Clinical Trials Planning Meeting for neuroendocrine tumors, and it really helped determine what the priorities were in neuroendocrine tumor clinical trial development. And progression-free survival, which refers to how long does it take for the tumor to grow, is really thought to be one of the best endpoints for neuroendocrine tumors. Though some of the other ones being looked at are response rate, so that refers to how much does a tumor shrink, and also overall survival. How long do people live if they get this new intervention? As you can imagine, overall survival is actually really hard to look at in this disease. It's unique for this disease because patients live longer than patients with other types of cancers and go on to get many subsequent treatments. So it makes overall survival sort of a muddy endpoint because of all of the other treatments that happen after that particular clinical trial is done. So you've seen a few of these curves in mice and in humans already today. I want to just explain how you look at these. So this is called a Kaplan-Meier survival curve. Along the horizontal axis here is time, and along the vertical axis is survival probability. So the percent of patients either alive or percent of patients who are progression-free. And then over time, you can see that this is an example about patients in remission. So over time, fewer and fewer patients are in remission. And same thing with this arm. In blue, this particular group does better, as you can see, at any given amount of time. So this unit, if it were 10 years, there are more people alive on the blue line than in the red line. So that's how you read those curves. So there are also a number of sources of clinical trials. So there are investigator-initiated trials. So this is when, Dr. Nakakura or Dr. Bergsland might go pitch an idea to a company and say, hey, I've got a great idea, will you support this? They're often smaller. They're usually available just at a single institution. The idea is initiated by an academic MD, and funding or drug supply is from the company. Then there are also industry-initiated clinical trials. These are often quite a bit bigger, where a company may say, we have an idea, but we need academic institutions to enroll patients, and they'll farm it out to a number of different institutions. Usually involves many sites, often international. So for example, the PRRT study is an industry-initiated study. As you heard, there are many sites in Europe and in the US. Funding is from a pharmaceutical company. And then third, there are cooperative group trials. So cooperative groups are actually federally funded or federally organized groups that really bring together academic institutions. So interestingly, Stanford is part of the Eastern Cooperative Oncology Group. That's sort of historical, though we are on the West Coast. UCSF is part of a cooperative group called the Alliance. It used to be CALGB, but there are a number of cooperative groups in the US, and we actually all work together. It's a perfect setting to do studies in a rare disease because it brings together a number of institutions. And then why are clinical trials important? I think this message has been relayed over and over again today, but it's really critical for advancing medical knowledge and patient care. We do clinical research to learn whether a new approach works well and is safe, and which treatments or strategies work best for certain diseases or certain types of people. And I think Dr. Fisher mentioned this earlier, very few adult patients with cancer actually participate in clinical trials. It's only 3%. So for those of you wearing your badges, we appreciate you for considering participating in a clinical trial, and I think that we certainly want to raise more awareness about it. Okay, so we'll move on in terms of talking about what we've learned from recent clinical trials. So amazingly, in the last, since 2009, I'm going to go through chronologically, we've had about 4 big phase 3 trials that have led to major practice-changing advances in this disease. So again, this is chronologic. So the PROMID study was a randomized phase 3 study looking at, it was fairly small, 85 patients with well-differentiated metastatic midgut neuroendocrine tumors. And they were randomized to receive either octreotide or placebo. And the survival curve, hopefully you now know how to read. So the yellow line were patients who received octreotide, and the blue line were patients who received placebo. The endpoint was time to progression, which is actually very similar to progression-free survival. And as you can see, it favored the octreotide arm. So those patients lived longer without progression. So a second big phase 3 clinical trial was called the RADIANT-3 study. So this was a phase 3 study of everolimus, which Dr. Nakakura just spoke about. It's an mTOR inhibitor. This was for advanced pancreatic neuroendocrine tumors. And as you can see, this was a much bigger study, 400 patients randomized to receive either everolimus or placebo. Here's that curve. So I hope you're seeing the trend. If the curves really have great separation, that is encouraging and very much suggests that those groups do differently. And as you can see here in blue, this study also favors the treatment arm with everolimus, and with the difference of 11 versus 4.6 months of progression-free survival. So again, this is not overall survival. This is the time it takes for patients to progress if they're taking everolimus versus placebo. So this led to the approval of everolimus or Afinitor in this disease. This was back in 2011. Very exciting. So a similar study was performed in advanced carcinoid tumors, 400 patients, very similar study design. You can see that there is some separation of the curves, though there are complicated statistics that go into this. So when the trial is being designed, they have to make a guess as to what the statistics will be around the p-value and how different those two lines will be. And this did not officially meet statistical significance. So I think many of us who treat neuroendocrine tumors think that this is an active drug in this disease, so it has not officially been proven so. RADIANT-4 is an ongoing study that will hopefully answer this question and is looking at, again, at everolimus in advanced carcinoid tumors. Fourth study, so this is looking at angiogenesis inhibitors. We've heard about this and you've seen a number of pictures today of very vascular liver lesions. And this approach in neuroendocrine tumors was thought to be very exciting. And so there was a phase 3 trial of sunitinib, which is an oral tyrosine kinase inhibitor. It's an angiogenesis inhibitor, looking at advanced pancreatic neuroendocrine tumors, 170 patients randomized to receive sunitinib or placebo. And this doesn't project quite as well, but the top line here is sunitinib versus placebo. So progression-free survival was longer in the sunitinib arm of 11 months versus 5.5 months. This also led to the FDA approval of this drug. So in 2011, we just amazingly had the approval of 2 new oral agents in pancreatic neuroendocrine tumors. This is a study that actually was presented for the first time last week at the North American Neuroendocrine Tumor Society meeting. It was the first US publication. I don't have pretty curves because I tried asking for them and they're not available yet. So, but this builds on the PROMID study. So as we know, octreotide in midgut neuroendocrine tumors slows down the growth of these tumors. But is that true for other non-pancreatic neuroendocrine tumor sites? And the answer is yes. And so this looked at lanreotide versus placebo. This was a study that included pancreas, midgut, hindgut, and unknown primaries. And I have the numbers for you, but not the curve. So this then compared, as I said, lanreotide versus placebo. Progression-free survival was the primary endpoint. At the time they did the analysis, which was about 24 months, they still had not reached the median progression-free survival for lanreotide, but they had for placebo, so 24 months and counting. So we know that patients on lanreotide had longer period prior to progression compared to placebo. So then moving on, we have some recently completed trials. Some of you in here have participated in them, and we're really eagerly awaiting the results. So these include also angiogenesis inhibitors, bevacizumab, that's an IV antibody against a protein on blood vessel surface, and one was in carcinoid and one was in pancreatic nets. So this was a SWOG study, that's a cooperative group study, looked at bevacizumab and octreotide versus interferon and octreotide. And the CALGB, this is the cooperative group that Dr. Bergsland's institution belongs to. And this was in advanced pancreatic nets, also looking at bevacizumab, but this time it was everolimus and octreotide versus everolimus, bevacizumab, and octreotide. So both of these have completed accrual, and we're just waiting for the trial results, which hopefully will be in the next year or so. So trials on the horizon, this is the exciting part. So a number of comments have also been made about what are the differences between some of these biologic or targeted therapies like Sutent, sunitinib, or everolimus and bevacizumab compared to real chemotherapies. So as was mentioned in one of the case discussions earlier, if we really want to shrink a tumor, we might consider giving chemotherapy. And so can we define a standard? Currently, streptozocin is the only FDA approved. It has a fair amount of toxicity. I'm leading a national clinical trial in low-grade advanced pancreatic neuroendocrine tumors looking just at this question of temozolomide versus temozolomide and capecitabine. They're both oral agents, very well tolerated. So this is about 140 patient study that we hope to finish in the next couple of years. What's next for carcinoid tumors? We mentioned this already. This is a study that is open, and as was the one that I just mentioned, Emily's leading this study. She talked about it a little bit earlier of tongue twister, pazopanib versus placebo in advanced progressive carcinoids. And then this was a study that we've spoken about many times today. I won't belabor it, but it's the lutetium study for PRRT. And then another theme that has come up today, what do we do post-operatively for patients? As Emily said earlier, there aren't standard options, but there's a new clinical trial that is also through the cooperative groups that are looking at patients with pancreatic neuroendocrine tumors who've had a resection of their primary and their liver metastases, and then randomizing them to everolimus versus placebo. So that's a whirlwind tour, but I think that it's incredible advances for a rare disease, much shorter than the timeline that Dr. Nakakura showed you from not a 40-year timeline that we've made major advances just in the last few years. So very briefly, I'll finish up with how can you learn about clinical trials? So I mentioned clinicaltrials.gov. I will say it's a little bit of a cumbersome website. It is searchable, but if you need advice, I would say talk to your physician about clinical trials that might be accessible to you. The Caring for Carcinoid Foundation also has a searchable tool to look for clinical trials. And then likewise, most cancer centers have searchable tools. So I'd encourage you to look at UCSF site or Stanford's or your local institution, but really talking to your doctor about what might be best for you. And these are just trials that are going on. We've talked about most of these already. So lastly, I'll finish up with the Neuroendocrine Tumor Registry. This is an effort that was really jump-started by a grant from the Caring for Carcinoid Foundation back in 2009. And the Neuroendocrine Tumor Registry is something that allows us to collect clinical information, tissue and blood on patients with all types of neuroendocrine tumors. We're doing it at Stanford, and many other institutions have their own databases. And in terms of the logistics of this, we have an online consent and questionnaire. Patients can do it either in clinic or at home. The blood and tissue piece is optional. It's, I think, as many have mentioned, important to learn more about the epidemiology of this disease. So as I'd mentioned, in 2009, we received a grant. We worked on database development and optimization of data collection. We have over 200 patients now. One of a medical student that's been working with me, Hillary Lynn, had a poster looking at pathology discrepancies in neuroendocrine tumors. That was the first project out of this particular database. So I think to finish, this is the last slide, I hope I leave you with really a lot of optimism about the research that's going on in clinical trials. There's been an incredible renaissance in this field, numerous active and developing clinical trials. Participation in clinical trials is really what moves the field forward. And I want to thank you as patients and clinical trial participants. So I'll stop there and happy to take a couple of questions. Any questions? I
have two questions, one for Dr. Nakakura. Is there a timeline for the INK-128 moving from laboratory mice to human beings?
That's a good question.
Not 40 years.
Yeah, not 40 years.
Not 4 years. Okay.
No, the important thing is I think this drug's being tested, I think, in Europe for various diseases. So once, I could maybe ask the oncologist, like Dr. Kunz, once there is evidence in a preclinical setting, like in a xenograft model, that a particular therapy is effective, how hard or how long does it take to get to actual therapies in patients, or do you have to then repeat the study in humans? So, for example, using this new INK-128 in patients that progress on everolimus, would you have to do a clinical trial, or could you perhaps introduce it into the clinic in patients that are progressing if it's shown to be working in xenografts?
Do a clinical trial. I don't know if this is on. So it would still need to be evaluated in terms of safety. So it would still have to go through, every drug that gets FDA approved has to go through phase 1, 2, and 3 clinical trial. So.
Right. If it's undergone that for other cancers in, say, Europe, would you have to repeat those all here as well?
Yes.
Okay.
The safety piece maybe, and I don't know, I might defer to George. The safety piece, no, but the efficacy piece in a new tumor type, you might need to. Yeah.
Okay. And I have a question for you, Dr. Kunz. There was a great poster presentation on CD47, and everything has been done using PNET on mice, but yet the decision moving forward is going to be on leukemia. Just curious how the decision process works.
Yeah. Is Dr. Krampitz in the room? He may have left already. I might defer that one to you.
So it's so that may apply to numerous different tumor types, and we're going to test it first in leukemia and in colon cancer, believe it or not, colon cancer and other solid tumors. Now, every once in a while, we can sneak a neuroendocrine tumor in there, but certainly neuroendocrine tumor is on the list. And so the advantage is that we will be doing that with Oxford University in England, and those will be the only 2 centers. So we'll be able to sneak some patients in with neuroendocrine tumors, I think.
Yeah, but yet the baseline was based on PNET.
No. That was just the poster, because this is a NET conference.
Okay.
Most of the data are for other solid tumors.
I'm going to ask a question of George. It's a question I've actually been asked a lot today, and I think might be worth addressing, is about immunotherapy. So, Dr. Fisher, does immunotherapy have a role in neuroendocrine tumors?
Not yet. So no is the easy answer, and it's you'll hear more and more over the next year or two, I think, of some promising work that's being done in immunotherapy. I think that every day, every full day of patients, and I'm sure the same is true with Emily, someone will ask, well, can I just get the vaccine for this cancer? And we're nowhere there yet. But I think that we're starting to learn about the immune system in such a way that we'll have tools to tweak it. And if we tweak it just right, I think that we might be able to get immune responses to a number of cancers, and hopefully neuroendocrine included. We're having a number of meetings that are devoted to trying to develop an immune strategy for neuroendocrine tumors, and that's just starting. Yes?
This is more of a basic than a translational research question, but I'm curious, given the fact that the neuroendocrine tumors are distinguished by their slow growth rate, is the reason that they grow so slowly well understood? And if not, is anyone researching that question?
You're stuck at the podium.
Yeah, sorry, Eric, I don't know if you want to answer that. You're welcome to.
Well, I think the very basic questions of what makes neuroendocrine tumors grow is a critical question. And I think once we start answering those questions, and I think some of the encouraging early results that Laura mentioned might help shed light on that. But we don't even know what makes them grow. And I think, to answer your question, compared to some more aggressive cancers like pancreatic adenocarcinoma, we've already identified a lot of the mutations that drive, or oncogenes that drive the tumor and make it grow fast. We're just starting to uncover what those genes are in neuroendocrine tumors. So I think that's the first question.
No, it's actually a critical question, and it does denote a different biology, and we haven't quite have come to grips with what are the molecular basis for those differences. Why some tumors grow like at a glacial pace and other tumors grow fast. So it's a very good question. I don't think we have an answer, and I hope that animal models will be able to give us some insight as to the growth rate and what determines growth rates.
Thank you.
For Dr. Kunz, looking ahead, are you seeing any time soon that there might be a PRRT phase 3 trial for us with pancreatic NETs?
So that's also a great question. I think we're hoping that the PRRT trial in advanced carcinoid will just open that door in the U.S. I think that I'm hopeful for that. I don't know in what timeline that will happen.
So that you don't feel neglected, it wasn't that we didn't consider it. The problem is that there are multiple treatments in the United States that are approved and effective for PNETs. And therefore, we'd have to have a control arm against one of those treatments. And it was going to be a little bit of a tougher sell and perhaps a higher bar than carcinoid. So we're hoping we're picking a low bar so we can get the drug approved, the treatment approved, and then move on from there. But it's a very good question. Bill?
Yes. Everolimus, is that affinator or affinitor, or is it the other one?
That's
Sutent?
Afinitor. So Sutent is the same as sunitinib, and everolimus or everolimus or potato, however you want to say it, is Afinitor.
Thank you.
We talk in oncology speak. Every drug has 3 names. It has a bunch of letters and numbers before it has a name. Then it has a name that you can't pronounce. And then it has a name that's easy to pronounce that the marketing people use. And so sometimes we use all 3 and it gets confusing.
One other sort of oncology dictionary tip is that anything that ends in an AB, so like bevacizumab, is an antibody. And anything that ends in an IB is a tyrosine kinase inhibitor, which is a sort of enzyme inhibitor, usually oral.
Yes.
I'm sorry I didn't bring it with me, but I saw a paper that was an in vitro study where they had done some kind of large array of cancer cells of different types, and they had tried a lot of old drugs to see if any of them would affect these different kinds of cancers. And they found an old tricyclic antidepressant. Did you see that one? With
Yeah.
Yes,
phenipramine?
it's actually the research originated out of Stanford. I don't know if you want to comment or.
So there's an old antidepressant called disipramine, which had too many side effects, and when Prozac came along, it blew it out of the water. So disipramine had some activity in small cell lung cancer and other poorly differentiated neuroendocrine tumors. And so Stanford has a trial looking at disipramine for people with poorly differentiated neuroendocrine tumors. So you're exactly right. And again, what works in mice or what works in paper, we have to prove whether it works in people. And so that trial is just underway. You're reading a lot. That's very good.
Yes, we should probably move on.
Yeah, so I want to thank the panel for the stimulating discussion.